So, dear students, I hope all of you are back and you're having much higher levels of energy, and we have to preserve that energy for the next 5 days, including this. So, before we begin the main schedule, I would have a small surprise even for you. So, having much higher levels of energy, and we have to preserve that for the next 5 days, including this. So, before we begin the main schedule, I would have a small surprise even for you. So, having much higher, we have to preserve that for the next 5 days, including this. So, before we begin the main schedule, I would have a small Civil Supplies Corporation and Finance Officer at the Bar Council of India. Her expertise is in budget planning, financial analysis, strategic resource allocation, and she has been very instrumental in driving fiscal success across multiple departments. She is also a guest faculty at prestigious institutions like the Bihar Institute of Public Administration and the RCVP Norona Academy, and there she trains the future leaders. So before I invite her for the speech, I would request Professor Graham Ball to kindly felicitate Mrs. Sandhya Srivastav with a flower bouquet. A big round of applause. Future leaders. So before I invite her, another round of applause. Thank you, ma'am, for coming, and thank you, Professor Graham. So now the stage is yours, ma'am. So very few words of motivation from your side.
Thanks to all of you. Thanks, Professor Graham, who has just felicitated me. Thanks to all administration of Allahabad University, who has given me this opportunity that I can interact with future leaders of this country. Definitely, you are very bright students from various parts of our country. And I'm looking that our country is very much safe in your hands. And really, I'm very much grateful for this organization, for this opportunity. Thanks, Professor Graham Ryle. Thank you, Professor Sanjeev Shrivastava. Thanks, Dr. Aru, who has just introduced me. I'm really very much grateful. I want to share a few words with my journey. Earlier I was also just like you, I was also a PhD student. I joined research, but after that I selected in competitive examination in Public Service Commission, and I joined as finance officer very long back. And after completing 38 years of long journey, serving in various departments, I came here. I want to share a few things which I find in my whole journey and which comes always with the experience. First thing which I want to say you, that is make your goal very clear. Your vision should be very clear. You ask yourself, close your eyes and ask yourself, God is within you, nowhere you have to search it. In Sanskrit it is told, Aham Brahmasmi, Tattvamasi. That is, God means energy. You can give any name of that energy. That is everywhere in the universe. You should ask yourself what I want to do really. What is my interest area? What I can do? Make a realistic, achievable goal. Dr. APJ Abdul Kalam has told, सपने वो नहीं हैं जो बंद आंखों से रात में देखे जाते हैं, सपने वो हैं जो खुली आंखों से दिन में देखे जाते हैं। सपने देखो, सपनों को पूरा करने के लिए पूरी जी जान लगा दो। और जब आपका विजन क्लियर होगा, जब आपका लक्ष्य स्पष्ट होगा, तब आपको दिशा मालूम है। लाइक दैट, व्हेन यू आर जस्ट स्टैंडिंग ऑन ए रोड and you have to see, if you are looking a clear path, you know where you have to go. If you don't have that vision, you cannot go very focused. अर्जुन की तरह केवल चिड़िया की आंख दिखनी चाहिए। जहाँ आप बैठे हो वहाँ सफलता का अर्थ कुछ और है, जहाँ मैं खड़ी हूँ वहाँ सफलता कुछ और है, लेकिन अल्टीमेट सफलता एक ही है। जहाँ आप खड़े हो वहाँ अपने आप को मजबूत करना, अपना कैरियर बनाना और कैरियर में आगे बढ़ना, ये सफलता की परिभाषा है। जहाँ मैं खड़ी हूँ वहाँ आनंद, प्लेजर, स्पिरिचुअलिटी, ऑल दीज थिंग्स ऑफ द सक्सेस। तो आपको सबसे पहले अपना गोल डिसाइड करना है। यू हैव टू डिसाइड योर गोल, क्लोज योर आईज, सिट एंड आस्क योरसेल्फ व्हाट आई कैन डू, व्हाट रियली आई वांट टू डू। Don't go just if people are doing this, so I have to do this. No. Ask yourself. When your goal is decided, सतत परिश्रम, दृढ़ आत्मविश्वास, आशावादी दृष्टिकोण. These three keywords which I find in my life. There are not every time we are getting success. Life is for the success and failure, criticism and praise, and always you have to face lot of struggle in the life. But one more thing which is very important, that you have to keep patience. Keep patience. हमेशा धैर्य रखिए। तो पहली चीज, आप अपना लक्ष्य निर्धारित करिए, अपने लक्ष्य को विजुलाइज करिए। Your thoughts give immense energy to you. आपके विचारों में बहुत शक्ति है। और every time you listen positive thinking, that is आशावादी दृष्टिकोण. But what is positive thinking? How you can think positive? What is the meaning of positive thinking? कैसे आपको negative thought के chain को break करना है? Every time some negative thought comes, sometimes कुछ आएगा कि मैं नहीं कर पाऊंगा, लेकिन आपको उस negative thought के chain को break करना है। You have to maintain, just give 5 minutes every day to yourself, not more than 5 minutes. 5 minutes is enough for you, at least 5 minutes. Just sit with yourself and in the morning, energize yourself, pay your gratitude to the universe and forgive to everyone. These three key points which I find in my life. And when you are doing, you don't do it for others, you do yourself. You have to do for yourself. संस्कृत में सुना होगा, तमसो माँ ज्योतिर्गमय, असतो माँ सद्गमय, मृत्योर माँ अमृतम गमय। आपको अंधकार से प्रकाश की ओर जाना है। विवेकानंद ने कहा था, उठो जागो और ज्ञान की ओर बढ़ो, उत्तिष्ठत जागृत प्राप्य वरान्य बोधक। उठो जागो और आगे बढ़ो। तो आपको सबसे पहले अपने आप को जानना है। बिफोर यू गो डीप इनटू दिस साइंस, you should know yourself, how much energetic you are, and you can do whatever you want. Really, you can do all. You have tremendous energy in yourself. प्रसाद की 2 लाइनें हैं, अमर्त्य वीर पुत्र हो, दृढ़ प्रतिज्ञ सोच लो, प्रशस्त पुण्य पंथ है, बढ़े चलो बढ़े चलो। असंख्य कीर्ति रश्मियां विकीर्ण दिव्यदा हसी, सपूत मातृभूमि के रुको न शूर साहसी। अराहति सैन्य सिंधु में सुवार्णवाग्नि से जलो, प्रवीर हो जयी बनो, बढ़े चलो बढ़े चलो। निराला ने भी कहा था, योग्य जन जीता है, पश्चिम की उक्ति नहीं, गीता है जागो फिर एक बार। या तो गीता पे जाइए या कठोपनिषद पे जाइए, जहाँ कहा गया है क्षुरस्य धारा निशिता दुरत्या, कि ज्ञान का मार्ग छुरे के धार के समान कठिन है, लेकिन चलो मजबूत पांव से, एंड यू विल गेट सक्सेस इन योर लाइफ। जस्ट वन थॉट यू शुड कम, आई शैल गेट सक्सेस, आई एम बॉर्न टू बी सक्सेस, आई एम सक्सेसफुल। विजुअलाइज योर थॉट। आंख बंद करो और बंद आंखों से अपने विचारों को देखो, अपने आप को एनर्जाइज करो, गॉड को, यूनिवर्स को, सबको थैंक्स दो, और थैंक्स के बाद हर उस चीज को माफ करो जो तुम्हें परेशान कर रही है, जो तुम्हारे अंदर निगेटिविटी भर रही है। दूसरों के लिए नहीं, अपने आप के लिए। ये क्लस्टर साफ करना है, जला साफ करना है। लाइक दैट, जैसे आप अपने घर से कचरा साफ करते हो। एवरी डे गिव 5 मिनट्स ऑन योरसेल्फ। अपने आप को एनर्जी दो, सेंस ब्रेन तो अपने मस्तिष्क से लेकर अंगूठे तक को, हर पार्ट को एनर्जाइज करो, और अपने आप को जो है, अपने विचार को एक बार दोहराओ, क्लियर देखो अपने विजन को, और लग जाओ अपने काम पे। निरंतर परिश्रम के अलावा, हार्ड लेबर के अलावा कोई दूसरा रास्ता मैंने तो जिंदगी में नहीं पाया, और मैं समझती हूँ ये सभी को गुजरना पड़ेगा जिनको बहुत ऊंचाइयों तक जाना है। और हमेशा चलते रहो, बिना थके बिना रुके। एक मुबारक सिद्दीकी की भी एक गज़ल की 2 लाइनें हैं, मेरे दिल के बड़े करीब हैं, मैं आपके साथ साझा करना चाहती हूँ। हवा की ज़द पे दिया जलाना, जला के रखना कमाल ये है। हवा की ज़द, तेज प्रवाह, हवा के तेज झोंके के बीच में, हवा की ज़द पे दिया जलाना, जला के रखना कमाल ये है। खिज़ां के रुत में गुलाब लहजा, बना के रखना कमाल ये है। विपरीत परिस्थितियों में भी, पतझड़ के मौसम में भी मिठास और जिंदगी में आशावादी दृष्टिकोण। तुम्हारी आंखों में चमक, तुम्हारे होठों पे मुस्कुराहट कभी गायब नहीं हो। खिज़ां के रुत में गुलाब लहजा, बना के रखना कमाल ये है। किसी की राह से हटा के पत्थर उठा के कांटे, किसी की राह से हटा के पत्थर उठा के कांटे, फिर उसके आगे अदब से सर को झुका के रखना कमाल ये है। फिर उसके आगे अदब से सर को झुका के रखना। हमेशा विनम्र रहो, हमेशा एटिकेट्स में रहो, हमेशा बड़ों की इज्जत करो। बहुत सारा जो कुछ बचपन में सीखा था, फॉलो इन द लाइफ एंड यू विल फाइंड most important thing, last but not least, you must be very conscious about your health. Not only physical health, emotional health, mental health, all it is very much necessary. बहुत सारे destructive force आएंगे, like अभी तो मीडिया बहुत ज़्यादा सक्रिय है। Sometimes you will feel that there is some criticism can be disturb you, but every day I have told you, sit in the morning and forgive to everyone, give energy to yourself, and thanks to universe, pay your gratitude. So you will go with the mental health, few minutes for yourself, for your physical health, mental health, emotional health, and then you will definitely get success. I can give you guarantee of the success. What I could get in my whole life, just I shared with you within few minutes. People have given me just few minutes just to introduce you. I am very much thankful to Dr. Arup and this university administration, Honorable Vice Chancellor, and to everyone that they have given just this opportunity to talk with you. I am grateful to all of you. You listen me very patiently. I am thankful to you. Lot of thanks, lot of best wishes, and lot of blessings to you. Go ahead, always go ahead. Thank you. Thanks. Your path is very bright. This is dream for several students to come here in this institution. You got it. Really were very much successful.
So, thank you so much, ma'am, and for your inspiring words, the way you spoke. I think that's a skill itself, how to speak on a public platform. So, and your message has truly motivated us, and it has resonated. I hope the entire audience would take inspiration from your words. And with that powerful motivational lecture, we will start off with our first lecture. And before we start off, let me introduce you to my mentor and friend, Professor Sanjeev Srivastava. So, Professor Sanjeev is really a luminary in the field of proteomics. He did his PhD from the University of Alberta in Canada, and subsequently he completed his postdoctoral training at Harvard Institute of Proteomics at Harvard Medical School in the year 2009. Thereafter, he joined the Department of Biosciences and Bioengineering at IIT Bombay in 2009, and he was one of the very first people in India who took up proteomics in the scale that you are seeing it around the world. So if you have to have a glimpse of what world proteomics look like, maybe you can visit Professor Sanjeeva's lab, and he's still doing that because he's recently set up India's first 4D omics facility at the company that he has founded, Proteomica International Private Limited. So he has started India's first 4D omics facility. And books, and more than 20 patents have been filed from the biomarkers he have discovered. He has also initiated the Cancer Moonshot India program, where he brought India into the International Cancer Proteogenome Consortium, and he has also contributed a lot, and he has been awarded profusely, especially like the Young Scientist Footstep Awards that he received in Canada, as well as the Apple Research Technology Award that he has received in UK. He is also the Fellow of the Royal Society of Biology, the Royal Society of Chemistry, and the Royal Society of Medicine. Beyond his research, he is a very distinguished educator, and he is leading the proteomics education and training programs in India. So if you look up about Sir on NPTEL, you will see multiple videos and lectures, and Sir regularly conducts training programs at IIT Bombay. In fact, that was the very first time we met was at IIT Bombay when I attended one of his training programs. So a very distinguished privilege that I could bring his expertise to my workplace at Allahabad, which I joined a couple of years ago. He also serves at the Council of Human Proteome Organization, HUPO, and the Executive Committee of the Biology-Driven Disease-Driven Human Proteome Project. So please join me with a huge round of applause in welcoming Professor Sanjeev Srivastava.
Alright, good morning everybody. While we're setting the projector, so let me just go a step back and where previous speaker left you. I think in your PhD journey, I think most of you are probably aspirant of doing PhD or research scholars somewhere. I think you'll always find, it seems very exciting when you join PhD program, whether within India or anywhere when you go abroad. But what actually becomes after that, the first 2 years becomes really challenging, right? Anywhere you go, because immediately expectation is like, you were trained in the small pond and now you have been left in the big ocean independently, right? You have to now swim and survive. Expectation is that you know everything now and you have to be handling the project independently. So that is the time when I think your actual, not only your hard work, but also your ability to cope up the stress is going to really dictate your success, right? So I think the previous motivation lecture was very relevant for you to think about how to not only do good research and good hard work, but also keep up the good health, both physical and mental, and have the patience and perseverance. Those are the skills which I think will take you a really long way. PhD is not a small degree program which you just being measured with your few coursework and credit program. It's actually going to be taking you in a very long journey And the first 2-3 years are very, very difficult because most of the time you start excitement, you suddenly see the excitement goes down. Then you will start thinking quitting programs. Many think that okay, it was probably not meant for me. But again, like think about your goal for which you plan to join the PhD, and the much bigger vision, much bigger picture. Within that, then the small problems will move away from your road, and you will succeed in that way. I think everybody goes through that that cycle. It's a good idea for you to really prepare yourself very well in this direction. So let me start with this small clip, which is one of the first big project happened in biology area, which I think in front of most of us, which was being announced in White House. So let's start with this video clip first.
We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind. The moment we are here to witness was brought about through brilliant and painstaking work of scientists all over the world, including many men and women here today. It was not even 50 years ago that a young Englishman named Crick and a brash, even younger American named Watson first discovered the elegant structure of our genetic code. Dr. Watson, the way you announced your discovery in the journal Nature was one of the great understatements of all time. This structure has novel features which are of considerable biological interest. Thank you, sir. How far we have come since that day. In the intervening years, we have pooled the combined wisdom of biology, chemistry, physics, engineering, mathematics, and computer science. Tapped the great strengths and insights of the public and private sectors. More than 1,000 researchers across 6 nations have revealed nearly all 3 billion letters of our miraculous genetic code. I congratulate all of you on this stunning and humbling achievement.
Alright, so this was actually one of the first big announcements made at White House, when first time we thought that we can now understand all the secrets of human life, and first time all the human genes were sequenced, published in 2 different parallel projects with the both public and privately funded published in Nature and Science. And this was the first time which we also learned that we are not very different than chimpanzee. We are not very different, in fact, than many other model organisms, right? So, like for example, C. elegans, yeast, Drosophila, we are all having the gene numbers, in the ballpark of around 20-25,000 genes. So immediately the entire focus shifted that how then we are so different than many of these organisms, and what makes us different. So it means in the central dogma of life, some molecules are much more important, which gives us that ability and intelligence and different phenotype. And immediately after the genome sequencing projects, the focus shifted to the proteomics and field of understanding the big omics sciences. So with this, let me start my first lecture, and let me first thank Dr. Arup for inviting me here in this GIAN program. I'm sure you're going to have a very stimulating experience in the next few days, but it will all depend on how interactive you make it. And it should not be just one way. It should, be very open discussion, because utilize this platform to really clear up your doubts and interact more with the speakers and your resource people here. So I thought, while I'm more specialized in the proteomics, metabolomics, and other areas of functional biology, but I thought to start with a little bit basics of genomics, because that was the foundation from which the entire big data sciences and the omics sciences actually started with. And then where we are currently heading with all this knowledge sphere of omics, moving toward personalized medicine. So I come from IIT Bombay, which I think some of you are aware of. It is one of the India's higher institute for research and excellence, especially institution of eminence status we have. You are welcome to come to some of the program and training which we conduct. I think I'll give some update toward the end. I come from Department of Biosciences and Bioengineering, where there's a lot of focus on the post-grad education and research. Almost 200 research scholars to 50 are doing PhD, and very strong program in the interdisciplinary research area. I established this national facility for doing the proteomics research and training program at IIT Bombay, which is again funded by Government of India. You're welcome to come and actually you're entitled to send the samples and analyze and make use of these resources, which we very much take from all external users. This facility is equipped with various type of Orbitraps, and we regularly conduct some training workshops which can be really useful for you. Given the pace of how the proteome should be analyzed in much faster ways one could do the sequencing, we have recently set up this new technology, which is the timsTOF based mass spec for doing the 4-dimension separation and 4D omics at our startup, which is Organised Proteomics and AI Private Limited, which was just inaugurated last week. And that will give us now much more ability of sequencing the entire proteome at very unprecedented rate, which was not possible earlier. Even we are very surprised, like how fast you can do these experiments. Typically, we have estimate of human genes around 20,000 human genes, what we have. And earlier it was thought about around 25 to 26,000 genes, but after pseudo genes and all were dropped, then finally 20,300 some genes we have. But from those, how many actual protein we have, there is no clear estimate. One would estimate that, okay, at least there should be 20,000 protein directly which is formed from the genes. But eventually with all type of alternative splicing forms and the post-translational modification, one would expect maybe million forms of these functional proteins. How to really sequence these proteins at the fast pace and look at in totality. So that's where the complexity comes. Unlike the DNA with the 480 GC letters, now when we talk about 20 amino acids and side chain chemistry, it's not straightforward. Not one method, one technology can really do everything in a very, very rapid manner. So that's where I think there's a lot of advent of new technologies which are taking place. And I think in my first 2 lectures I will try to walk you through about where we started and where we are currently stands for, and where the more opportunity lies in the future. So my lab does a lot of work into different technology platform and assay development, different type of software and databases which we develop for the public, and also we have done a lot of work in the brain tumors and other cancers, and some work on the infectious disease area. But again, as I said, I'm going to make it more basic lecture, and aim is to give you GIAN in not the research topic to tell what we have done, but rather the basics of the technologies and tell you what you can do from these. So genomics started with looking at the ambitious goal of sequencing all the genes. Again, that became very first effort of looking at a big biology project in a very systematic manner by 15 countries together, with thousands of scientists coming together, with billions of dollars being invested. Goal was only simple to look at the sequence of one human, right? But then after that, then we realized that okay, we need to really study the gene expression, looking at the protein expression. That is going to dictate the biology, right? So that's just how I'm going to talk to you about some of the technologies in this space of genomics, moving to transcriptomics, proteomics, and multiomics. So genomics project was really remarkable, gave us the blueprint of life, but that in a way turned out to be slightly disappointing than what was estimated. We really thought that we was going to unravel all the secrets of human life after the genome sequencing project, and that was the time when I was actually planning to start my PhD journey in 2001-2002. But I was very excited when I was working already samples on the proteins, and I was very excited that okay, now the proteins are in the forefront, because after genome, everybody realized that we are still a lot to learn for the complex human system. So proteomics is going to be the next data science field, which is going to give us more information about the secrets of life. So that's where I think then I chose my career. But again, genomics also gave us not only the first time the genome sequencing ability, but also the high throughput sequencing technologies, also the way to work on the big data sciences, and first time developing the databases for the human, which actually served out the reference genome even for the protein search. So that was not just the doing the genome search, it actually really impacted many different ways of our understanding. And this human genome project became one of the flagship projects of the entire human history, when the aim was to not only look at the human DNA or the sequences, but also look at the other model organisms, trying to sequence along with the human, and can we really identify all the stimulants of the genes and store them in a database form, make them publicly available because it is a publicly funded project. And also then we started realizing that big data biology will have its own legal, ethical issues, and how to really handle that, because now we are revealing everybody's secret of life uniquely, and are we going to make it public? So if you sequence the genome, how are you going to handle that? And are you going to really reveal the personal disease, the racial information, all kind of, because people have a lot of sentiments around that. So again, that also brought in many new aspects of understanding that biology is not alone. You have to think about many social aspects as well. So this first genome sequence project was actually done on the 20 individuals. One of them was from the blended ancestry, which was essentially American with some Asian. But this was again in any biology research, as you know, we keep the anonymous stuff, right? So we don't reveal what the patient ID is. So this was again anonymous, which individual were sequenced, and 19 were from the European ancestry. Together with that entire sequences, it was put together a reference genome map, which is what being used to search against all the human genes and all the human proteins. Idea was simple. Can we take every chromosome, cut those into the small pieces, clone each fragment, amplify them, and sequence those small fragments and put it together in a logical way so that now we can start getting the long reads. But it was the very beginning of the entire genome sequencing efforts, which again, if you look at timeline of biology project, it goes decades, right? So that's where you need a lot of patience to really see good fruit of your work. This project started in 1984, and 1989, Dr. James Watson, he became director for that project. Eventually, 1990, they started a 5-year plan for human genome sequencing. 1893, Francis Collins became director. 1994, they published first genetic linkage map. Eventually, then they came with the first pilot project of human sequencing. And 2000, you saw that it was actually announced that now we got the first draft human genome map, not the complete sequencing, but the draft genome map, which actually got published in 2001. And 2003, it was announced to have it completed. One scientist which made huge contribution to this was Craig Venter, which along with the publicly funded project, they did privately funded. And this was really, very fast track, which they finished in a very almost similar time than the public funded, and both published same year, Nature and Science. So why this project was very, a milestone, because not only it gave us just the sequences of human gene, but also in many different ways of our ability to think about our environment, our health, and many other aspects which is now dictating, and now we can realize that the long-term impact of those has reached to precision medicine, looking at different type of the treatment therapy, right? Everything is being dictated with those just knowing at the sequence of these model organisms. And immediately, as soon as we got the genome sequences, now landmark studies started coming in. For example, we had prediction about human evolution, but there was no credential evidences made. But now, in 2010, they sequenced the Neanderthal genome and found that, it was probably the closest ancestor. So now there's a credential evidence made. In 2009, again, there was a big study made on the brain tumors, glioblastoma. They sequenced all these patients suffering from the gliomas and glioblastoma, and first time identified a very commonly known protein, isosaccharic dehydrogenase, IDH, which has a lot of mutation. And that became a landmark, now one of the key target for classifying these tumors. So again, our ability of looking at different aspects of biology, from evolution to medicine, has totally, truly transformed with just this basic discovery. So how the sequencing was performed in a very crude way, basic way. The Sanger DNA sequencing was started when they were looking at very, very short stretch of DNA sequences and then trying to put together, which helped us to first do the very small genome sequencing of the bacteria, Haemophilus influenza, 1995, moved with very other model organisms from the East, Drosophila, Arabidopsis, human, and eventually mouse was even after human, got completed. Then this concept of Sanger was very difficult, so then they took, can we bypass the chain termination method and use now the next generation sequencing. And this is one of the technology, one of the area genomic sequencing, where you see last 2 decades, we have really proved all the predictions wrong, right? The pace at which now the genome can be sequenced, it took 15 years time for first genome sequence project to complete. Now it may take, maybe less than few hours for one genome sequence to complete. It took $1 billion to do that project. Now you go to any institute and in just, maybe less than 1 lakh rupee, you can do the entire genome sequencing. So think about the pace and the precision and accuracy. This is where the engineering discipline, interdisciplinarity has really contributed a lot, right? And we can see that how this technology has really surpassed the Moore's law and all the predictions. So again, looking at the next generation sequencing, the idea was, can we make utilize our different physical principle, how these ATGC bases when they are going to release, release the inorganic phosphate molecule, and based on that, light can be now sequenced when the ATP, TTP, GTP or CTP, when they are binding, a flash of light is being utilized to sequence. But again, idea was this was a very iterative process. Can we now do much more high throughput manner? And then now the third generation sequencing technologies came. And therefore now idea was, can we utilize the entire stretch of DNA for sequencing? Can we take double stranded DNA, denature it, make single stranded, let it pass through some ways for doing sequencing, and now a lot of physicists, engineers came in forward and started utilizing what is the difference between ATGC. Can we look at their physical property and try to map these base pairs? And now one of the technologies is nanopore sequencing. It just looks like in the membrane, porous space, how the ATGC, when they pass, they have different perturbation. They do different type of electrical conductance change, and based on that, can you now measure ATGC in a very, very rapid manner, which is now being utilized with a very, very handheld device, a small device, which can be sent to the rural areas. Any place you go and take this small like hard disk type of the nanopore sequencing device, and then do the genome sequencing, send the data back to server, and now it can be it can do the genome analysis anywhere. This is just one of the technology. Of course, I'm not advocate of any technology, but giving you like how technologies are getting more miniaturized and robust. But much more specialization is coming from Illumina technology, which is one of the leaders in this area, and so as the Thermo Fisher, where they have iron current technology. We are not genomics lab, but often you have to correlate the proteomic changes with the genomic mutation and a lot of alteration at the genome level as well. So we also have one of the iron tolerant genome sequencing technology. In this one, I think one of my PhD students will show one of the setup we have to sequence
The instrument behind me is the Iron Torrent S5. This instrument detects the proton ions released during the formation of phosphodiester bond as a signal for sequencing. It comes with two other smaller instruments, which are Iron 1Touch2 and Ion Enrichment System. The first step in a sequencing reaction is library preparation. Once that is done, we use the Ion 1Touch2 system for clonal amplification of the library. After the library amplification, enrichment is performed on the ion enrichment system. It uses biotin and streptavidin interaction to select the template positive ISPs. The last step is to start the sequencing on the Ion 1Touch S5 instrument. For that, we load the sample onto the chip. The sample should be uniformly loaded on the chip, and then we place the chip on the chip clamp. We close the chip clamp, and then we check that the washing solution, cleaning solution and reagent cartridges are in place. After that, we start the sequencing.
So again, this is one of the very simple way of looking at genome sequencing for millions of base pairs, and with the massive sequencing one could achieve, but we do more for the targeted work in this area. Another technology which is very much gaining momentum and news is the CRISPR technology. You might have heard the last year, 2023, the first CRISPR technology for the sickle cell anemia got approved, right? And again, our ability of editing genome, sequencing genome, modulating genes is now started truly revolutionizing our ways of treating the diseases. Sickle cell anemia, looks very simple, but individuals who have the defect, their oxygen binding capacity is severely affected. And while the basic science research has identified there is some important gene in the BCL11 which is going to be very important to as a target which can be utilized, but our ability was not there to deliver it into the very, very right form of editing. So CRISPR came again with the basic knowledge, looking at the cluster of regularly interspaced short palindrome repeats, which is identified in bacteria. An idea was from this bacteria uses CRISPR for its own immune system, its own defense against the bacteriophage. When different viruses infect the bacteria tries to build its memory by adding some of these spacer sequences. So it's already had the repeat sequence fragments, but it adds, let's say, one bacteriophage came attack, it actually adds a spacer from the degradation product of that phage DNA. It added that DNA sequence in its own genome to add the memory of it. LICO is another phage infected bacteria, and bacteria cast protein, which is actually now killing the phage DNA. It's actually after that it brings the DNA into the bacterial genome and now makes it another part of the memory. So now next time when the phage infect bacteria, bacteria is better prepared to kill the phage. So it has actually utilized this particular system for its own benefit and advantage. But now scientists thought, okay, can we utilize the bacterial learning for the CRISPR-Cas system and utilize for the treatment? And that's something which got Nobel Prize to Dr. Emmanuel and Donda for doing the CRISPR-Cas technology in 2020 Nobel Prize. And that immediately started showing the big impact. Now we know, like in the human growth, when we have this protein for the sickle cell anemia, which is having the mutation, that happens like if you're a little bit familiar with the hemoglobin protein and the myoglobin protein, right? So in hemoglobin there are tetramer, alpha 2, beta 2. So one of the beta protein gene that gets just one base pair change, and because of that the protein totally becomes like sickle cell shaped. So in our childhood, a protein is for the hemoglobin known as HbF, hemoglobin fetal, and adult known as HbA, which is hemoglobin adult. Now that transformation from the fetal to adult is transformed with the transcription factor BCL11A. So now what people thought, can we utilize the BCL11A gene, because that is very efficient, because in the childhood inside the womb the embryo has to totally do the very efficient oxygen binding inside the mother. So that HbF is much higher binding capacity than HbA. So then people thought, can we now utilize this BCL gene and try to change the course of when at the time of birth HbF level goes down and HbA level goes up. Can we find out this BCL11 switch and try to control that, modulate that, so that now HbF level remains higher in these individuals, and that has led to the cure of sickle cell anemia, which is, I think, amazing, and is just the beginning of many of the remarkable discoveries which you are going to see in front of us when a lot of clinical already trials are going on for different diseases, and something which you will be witnessing more and more, how these understanding and editing technology are going to impact. There are many ongoing efforts in the genome. The first one which I showed you was about the 20 human sequences which were made as a reference genome. But think about it, did not have the Asian representation. So whatever you were searching in the genome, it was more about the Caucasian data. But now then they added 47 people and more recently 350 people. So again the genome references are getting enriched with variety of population. And more so, it is now every country started realizing the power of the genomics revolution and started launching their own program. And India is not much behind in that. Even we have the 10,000 individual genome sequence which already got completed in March, and more of the secrets are going to be out soon. Additionally, we have a national cancer bridge connected from Tata Memorial Hospital in Mumbai, which now connects all the different states and their nodal centers, and trying to collect all the digital information about the cancer patients, trying to utilize that information pool at one place, which is again, it was not possible in previous years. All the clinical research relies on clinical data, and such type of initiatives are very, very important. Kerala, being very progressive state, started actually in fact sequencing the populations which are more prone for sickle cell or malaria or dengue. And now they are doing these endemic populations in the Wyand and Attapadi, these provinces. They are looking at the genome of these individuals which are more prone, what makes them more prone for the getting infected with the dengue or sickle cell. And again, these are just, you see, like population-based screening at the genome-wide level, which is going to really transform the healthcare system. So again, many of these are just happening in front of us, and I think you're all part of it. But I thought to emphasize you where we started a big data sciences, the genomics, where we are leading with the new and new developments in the field, from the Sanger method to the NGS method and many type of new sequencing technology, from nanopore, Illumina to the ion torrent, etc. How now this information, which is the base reference map, is going to lead us towards understanding much more functional content. So again, the looking at from the DNA to the RNA form, and again you are all familiar with the reverse transcription PCR or real-time PCR and impact of that during the COVID time, when we only knew some of the sequences of the virus. From the patients, the mRNA was extracted, cDNA converted, and now that is sequence for the specific genes of the SARS-CoV-2, but same could be utilized for many other applications. Likewise, you can always look at where the gene is being expressed in a technique known as in situ hybridization, which is essentially very elegant way of utilizing microscopy and our understanding of different gene expression and making those probes, and then trying to see where the genes are really expressed. So in Drosophila model, they have shown to look at the developmental stages. If you have the probes for looking at different genes for the wingless and ingrailed genes, which is involved in the abdomen and the wing, where they are being expressed throughout, you can see very beautifully, like how the mRNA being expressed. And for more and more gene, you have more probes. You can see live, like where the gene is being expressed. So again, the C. elegans and zebrafish and these Drosophila are very nice model when you can visualize the gene expression in the live cells. Right? Gene expression in the high throughput manner is another thing which is being thought of. Can you look at entire genome-wide, proteome-wide expression of these genes, and the small chips on which we have thousands to millions of features printed, can you utilize them for looking at the gene expression profile. So idea is, if we have this gene microarray chip, if let's say you are comparing the cancer cell with the normal cell, you extract the RNA from the cell, tumor cell and the normal cell, you convert them to the cDNA, and in the process you label them with different type of fluorescent fluorophore, Cyanin 5 or Cy 3, and then you mix them together, and based on the principle of hybridization, if the DNA was printed on the chip and the cDNA is going to get bind to there, you are now going to see the expression of these genes. And what you see at the end, you are seeing some of the very colorful dots of green, blue, red, and the the gray. And what it tells, like because you have labeled with Cy 5, Cy 3, this is probably highly expressed in the diseased tissue. The red ones are showing, the green ones are showing you it's much more in the normal. It was down regulation in the actual disease condition, or both of them are showing the same pattern. It means it is going to be no specific expression for the disease, or the gene was not expressed. So again, just by looking at this profile, why it is so important? Because now first time we learned that breast cancer is not one cancer. Just if you look at just the gene expression profile, women who are having different type of hormonal receptors and these kind of unique gene expression, they make them 4 subtypes of luminal A, B, HER2 and basal light. And based on that, now we know that with estrogen receptor or the progesterone receptors, herceptin receptors, or all of them being absent in the triple negative breast cancer, that is very unique. And if you are giving the same chemotherapy to every woman suffering from this cancer, it's going to not respond same way. And then that's why we see so much deleterious side effects, right? So now if individuals can be sequenced beforehand, they will have the probability of understanding the disease ahead of time. Doctors can treat them much more personalized way. And again, like these sequencing technologies can be also truly revolutionary, and sometimes celebrities bring a little bit more knowledge to the public in a way which you will see in this clip.
Her face is known around the world, and now her secret is known too. Angelina Jolie revealed in a New York Times editorial Tuesday that she had a preventive double mastectomy earlier this year, after discovering she has a gene defect that greatly increases her chances of getting breast and ovarian cancer. She writes, "My doctors estimated that I had an 87% risk of breast cancer and a 50% risk of ovarian cancer, although the risk is different in the case of each woman." While Jolie's genetic risk was very high, cancer experts say it's also fairly rare.
So 5% of breast cancers in the United States occur in women who have the genetic mutation, the BRCA1 or the BRCA2 gene.
Doctors say women with a family history of breast or ovarian cancer at an early age are more likely to carry the mutation, and insurance companies usually pay for genetic testing. For those who test positive, Dr. Rachel Brem says having both breasts removed like Jolie did is a very personal and difficult decision. It should be made with the help of genetics and cancer specialists.
It doesn't mean that that's the right decision for everyone. And the important thing to do is to have frank discussions.
Jolie said she went public because she wanted women in her shoes to know they have options and can still look and feel like a woman after a mastectomy.
Well, I think one of the reasons that we're seeing an increase in what we call bilateral or mastectomy on both sides is that there are better reconstruction techniques.
The actress was treated in the California Breast Cancer Center.
We hope that the awareness she is raising around the world will save countless lives.
Jolie's star power is already drawing attention to a complicated choice some women must face. Kelly Dashel, Associated Press.
Alright, so again, looking at so her family history was her aunt and mother to perform those functions. So the body conceives those signals to where to do what exact activity to do, right? The genes are there, but they are going to do specific transcription, translation towards that activity. Therefore, measuring this dynamic perturbation of transcript and protein become much more crucial than just the static blueprint itself. And looking at the spatial, the cell type protein information, or looking at the temporal different time change protein, even circadian rhythm people are studying, all that is really crucial to really understand the physiology of a given system, right? So proteomics, what do we study in the proteomics? Now question to you. What all do you want to study in the proteomics? And what do you achieve, what do you aim to achieve from proteomics? Please raise some hands. As I said, it should not be one way, right? Because now it is a small group, more interactive.
I think those who are sitting at the back can come back.
No, everybody, we should give opportunity. I'm sure you know that. I've already given you a lot of context here. Based on this, why do we study proteome? So Dr. Arup is going to, he's well-trained at IIT Bombay. Now he's going to conduct some quizzes as well to understand your knowledge, how much you are able to absorb, right? And as I said, please make it more interactive, because that's the only way you will take something back, right? It should not be one way around. My lot of lectures are already on YouTube and GIAN and many other portals, including this IIM portal.
Sorry to interject. So we are having Professor Sanjeeva only for this session. He has lots of lectures filed up at IIT Bombay as well. So I think you better use this opportunity to interact with him. YouTube lectures are there, but you cannot talk with Professor Sanjeeva over a YouTube lecture on YouTube. Maybe we are a couple of decades back, maybe in 10 years time you will have that facility. But right now I think if you're raising your hand, yeah, please.
To understand the structure of protein and how proteins are organizing, what are the dynamics we are creating, and what are their folding statistics. So that is why I think
proteomics, we can also like study
the revolution which is witnessing together in the field of biology, and especially the new area which is coming up, already started coming many places, but especially India has to come to look at more personalized medicine. Again, now you've seen a couple of examples, but now if you think about every individual very unique, coming from the very unique family history, family background, dietary, the environment, and all of them makes every individual so unique that you can't treat for a given diabetes, given hypertension, given cancer, everybody the same way, right? And that comes like, very elegantly from this example of Lucas Wartman. He is a doctor, physician at Washington University in US. He was diagnosed with acute lymphoblastic leukemia, ALL, and he was being treated for the ALL the way every patient is being treated, but he was having a lot of, after chemotherapy, a lot of side effects. And he himself was a genomic scientist. So he requested university whether his genome can be sequenced. And given that he was not responding well and he was terminally getting ill, they allowed, university allowed, although it was not as a part of the, the treatment modalities for the US healthcare system. But university allowed him to do sequencing. After the sequencing, what came very unique, that he was having some mutation in a specific gene found for the renal cancer or the kidney cancer. There is a drug, sunitinib, which can actually inhibit this particular FLT3.
Relapsed leukemia. Was admitted into the hospital and started on very strong chemotherapy. Unfortunately though, the leukemia didn't go into remission after being treated with this very high-dose chemotherapy. There were no real promising treatments on the horizon, and so things looked rather bleak. But at the same time, as I was being treated, my leukemia genome was being sequenced by the Genome Institute here at Washington University School of Medicine. We got back the sequencing results which showed that there were promising targets that weren't even in the scope of our standard treatment protocols for this type of leukemia. Because of those results, I was able to start less aggressive chemotherapy that I could tolerate, and then targeted therapy that had been approved for actually renal cell kidney cancer. Within a few days, my blood counts improved, and within a few weeks, my leukemia was in remission. So it was really going from a situation that looked really bleak to now a situation where there was hope again.
Alright, this is one of the great example I see, because I followed his study in 2012 when he fought and doing research. New gene defects which can be controlled with different drugs very effectively if we know our precise information. And again, as I said, these kind of transformations started happening in many places, especially in US. Like in one of the Michigan Center for Translational Pathology, the way they do the patient, like on the day one, for example, let's say tumor biopsies are being done. Pathologists will take the sample for looking at the routine pathology images, and then the samples are being prepared for the RNA, DNA, protein, and then different type of sequencing methods are being used to analyze the data. Then the sequencing tumor board consists of these doctors, clinicians, and the omics data scientists. Everybody together looks at one individual in its uniqueness and look at what all structural rearrangements happened, different mutation happened, gene expression happened, and what should be the best treatment given to that individual. So now, in 1 month is almost required, 4 weeks after doing the surgical resection to recover anyways, and during that time we are providing the patient a very unique way of treatment. This is really transformative, and lot of work and lot of good publications are in that area. Again, like the way you are going to be more exposed about artificial intelligence, machine learning approaches is going to really help us to narrow down the choices for clinician. It can't replace clinician, but it's going to really make them better informed and give them better options to choose. Look at the different genetic disorders, even cancer subtype detection, predicting disease progression much ahead of time, and looking at analyzing genomic variants, etc. I'll give one more talk, but I may not have sufficient time to give you a lot of my research talks to get today as well. But just came to mind that during the COVID time, Arup was in our lab and Professor Graham was working from UK, and we were working collaboratively. One problem which we were looking at, that how different patients are, everybody's mild form, but some people are getting to severe form, right? So we were, the clinicians were very much in a difficult situation to whom to prioritize and give them the bed and do a ICU or the hospitalization, because everybody's panicked, and those who are more influential will get the bed. But rather than that, can we look at the markers, like who really needs the support, right? So they were looking at some indirect markers of looking at, the various type of biochemical features and looking at their IL-6 level, interleukin-6 level. Many of these things were giving them indirect information, but no credible test was being made. So we were working on the COVID-19 research, and Dr. Rup was part of the team, who was looking at the entire blood plasma proteome profile of these individuals. And the team identified the big signature of the protein, which is changing from the mild to the severe form. But now, which among those could be the actual target to really develop the test? And that's when Professor Graham came in touch, and he did machine learning to say where the major nodes are coming from these, the data cloud which we generated, right? And based on that, we identified 4 major proteins. Apolipo-B was one of those, and we have the fibrinogen gamma chain protein and serpin proteins. So based on these proteins together, we were able to now predict individual much ahead of time that how one could actually progress from the mild to the severe form. And that was the first test which got patented, which is uniquely able to make prediction about a COVID patient at the mild stage, maybe moving towards the progressive stage of the severe type. So again, the artificial intelligence machine learning models are going to give you the prediction ability much more accurately based on your data models. It cannot just happen just out of the room, but it has to be based on the credential data sets. Again, one will look for more non-invasive methods for cancer detection. Can we look from the breathing? Can we look from saliva? Can we look from our sweating? Variety of ways. One is thinking urine as of course another sample type. Blood is still invasive, but could be used for detection. Many of these sample types can we really do much more accurate ways of understanding disease and even looking at better cures, right? So I hope my first lecture gave you the, at least set up the stage for your entire GIAN program, how these various omics technologies have really, which is happening in front of us, around society, around your clinics, has really started giving us better ability of looking at every individual in much more detailed, in-depth manner, and everybody can get better, kind of disease prediction or better treatment by utilizing these technologies. And over the time, with more and more good models being created with artificial intelligence, probably our prediction ability will also keep improving, right? But you need a lot of actual data set to work in that area. With this, let me thank you for being very patient and having at least some good questions. I'll stop here. If you have any questions, please ask me. Questions, comments, anything which you want to share?
Good afternoon, sir.
Hello.
Sir, you told us about BCL11A gene, which has helped us for treating sickle cell anemia as it helps in hemoglobin switching. So, sir, may you please throw some light upon this technique of how this gene was used or manipulated in order to treat those patients?
That's right. So, very good. There are two different things that are happening here. One is, of course, the basic science research, which discovers the role of important proteins and important genes, right? Which people have done differently. Like, they were looking at hemoglobin expression and what makes the fetal different than the adult form. That actually led to the discovering of this particular transcription factor, which was expressed at the switch off from moving from the fetal to the adult form, right? But then we got the CRISPR-Cas technology, which gives us the better ability of doing the gene correction, gene editing. And since we know that in the beta gene of our complex of alpha 2 beta 2, that defect can lead us to this problem of the sickle cell anemia. So idea was, can we utilize this particular gene which makes the protein for very efficient way of oxygen binding in the embryonic stage of the fetal, and use that gene to correct the defect for the adult, right? Because this gene which makes the protein for the HbF is going to give very strong oxygen binding. So the two things were brought together with the treatment of this technology, but were discovered separately. So like that, the CRISPR is just an editing tool which is going to utilize many knowledge of different basic science labs for different diseases, but now how to deliver them very accurately and very precise manner, in corrections. Any more questions? Alright, Arup is not here, but I think we thought we'll give you 5 minutes break, just in case you want to stretch your leg and just want to come with a bio break. 5 minutes, then I'll have another lecture which will be a little higher level now, more on the proteomics side. So just grab some water so that you are not sleepy, and we'll start the second lecture. Alright, thank you.
OBS डिस्कनेक्ट हो जाएगा तो नहीं कर जाएगा वो अभी लाइव जा रहा होगा ना वहाँ पे लाइव जा रहा है
If you have any problems, you put it up the problems in the WhatsApp group, right? And yes, be more interactive because it's very difficult to get
एक लेयर और बना करके हम लोग ये डाल सकते हैं कि ये क्या नाम है हेलो
extremely busy.
सुनो
And we have found time for you. So we need best use of this opportunity. Don't let it go. And you never know when what strikes you. All of you are in the very early phase of your careers. Maybe you are MSc, first year of PhD. Even those who are in the final years of PhD could kind of talk about a postdoc position. So make use, and you never know where things go.
Don't be afraid to ask a question, and if there's such thing, it's a stupid question, because sometimes a question that comes from somebody that stimulates my mind process that leads to a whole new opening. So please ask questions, please make points, it's important.
And I'm constantly focusing on a particular pathways of lipid synthesis. But what I can see is that most of the drugs that have been described in literature till now aren't working the same on my species. So how can that treatment data show light over that? What are the changes that are myself? And why, what other motivations for cancer that you need to see?
There is nothing, good that comes insight from your research, which is in there. I think drug discovery is a very difficult process, right? And I think the good drug is always challenging.
इसका कुछ करिए सर डिजिटल इंडिया
That's what I think, I'm sure
सबका बंद है इधर ही बंद आएगा कनेक्शन.
But this is what you are saying, the real research problem, where you don't really spend a lot of time and then eventually after convert some cell culture experiment of 6 months time, then you will start from scratch.
कितनी देर बाद चालू करेंगे?
ऑलमोस्ट बस 12:40 हो गया. मैं जाऊँ पीछे. ये दोनों फोन यही रहेंगे ना?
ये दोनों फोन से.
ये वाईफाई के लिए और ये उसके लिए. और तो प्रॉब्लम है तो पूछा.
Be a muscle. Make a bone. Carry nerve signals. DNA tells our cells exactly what to do and when to do it. That's why DNA is often called the blueprint for life. But how do our genes give such orders? Each gene consists of a recipe for a certain protein. Proteins are the machines and workhorses of our body. Proteins trigger cells to perform specific tasks, enabling the cells in your eyes to function differently from the cells in your heart or brain.
Our bodies are made up of about 100 trillion cells, which each contain thousands of different proteins. The collection of proteins within a cell is called a proteome. The proteome is the counterpart of the genome. Our DNA remains the same throughout our lives, but our proteome changes. This explains how the grown-up flying butterfly can become something so distinct from the young fallen caterpillar it once was.
Few proteins work alone. They interact and form relationships with each other. And the nature of these interactions, you could say proteins have their unique fingerprints with a technology called mass spectrometry. This technique can determine the identity and, indirectly, the function of proteins.
The process involves shooting charged protein molecules or fragments thereof into a vacuum tube and measuring how long it takes for them to fly to the other end. This flight time depends directly on their mass. Another way we learn about the molecules is by making them collide with gas molecules, which causes them to fragment into smaller species. By measuring the mass of the fragments and roughly 1 million fragments. This meter shows the huge amount of data we are constantly producing by measuring proteins. Gigabyte after gigabyte. This information helps us to precisely identify proteins and the social networks in which they exist.
Unique to humans, every living species on Earth is built up of proteins. Jellyfish, polar bears, tulips, and even bacteria. For this reason, study of the proteome will ultimately lead to better knowledge, not only of diseases and the effectiveness of drugs, but also of how plants can adapt to dry or salty environments, and how we might eventually be able to combat bacterial resistance. Excitingly, in recent years, scientists have learned how to reprogram the proteome of skin cells in a lab and turn them into stem cells. In other words, scientists are now able to tell cells what to do. For instance, regenerate tissue or repair a damaged organ. New therapies for a healthier society are on the horizon due to the fascinating research of proteomics.
Alright, so welcome to made by Dr. Prayagraj and team from Hugo. So this talk I'm going to take you more focused on proteomics, and especially then I'll take you to the more advances being made in proteomics field. I'll try to give you the variety of tools, techniques, how we separate the complex proteome. Mass spectrometry has become one of the buzzwords. I'm sure some of you may have already used it. Some of you at least would have probably studied or have learned about it. But I'm sure everybody is very much aspiring to learn about mass spec-based proteomics. There are many novel proteomics applications which are being utilized within these technology platforms now. How now biology is really benefiting with these technologies. I'll try to give you some sense of that. And a couple of databases and software which is being made, and also we have contributed recently. So again, like when we are thinking about the proteome, we are now thinking about a very complex central dogma of life, right? Moving from the genes to different type of isoforms, to looking at different type of post-translational modification, the proteoforms. 20K has become 70K to more than 1000K to the million forms. And now one technique cannot utilize to give you all the answers, right? To capture this particular dynamic change. So for doing any type of proteomics based investigation, you always start with taking the cells in question and try to start extracting all the proteins from the given cell type. And there is no one technique which can be utilized, no one method can be utilized, no one buffer could be told you, no one sonication or any type of lysis method can be given to you. Depending on the nature of the cell, what you want to extract out of the cell, you have to try out different things for extracting all the proteins. Sometimes actually we don't want to get all the proteins from the globular part because you only want to look at surface proteins, or only membrane proteins, or only you want to look at the subcellular fractionation. So again, depends on the objective. You have to be, this is where you students and the researchers, your knowledge and biochemistry comes into the play, the first part. Many things which seem very fancy, which you actually are more keen to learn, they are very standard, they are like, right, once you have got good protein samples prepared, rest of the things can be done very, very mechanical way, right? So I think you should think more about your actual problem statement and how you can get good protein out, devoid of nucleic acid and the salt and lipids and carbohydrates. Every molecule is important, but for you they will be all the, every molecule is important, but for you they will be all the contaminants and the artifacts. One could use different ways of protein separation. I just showed you one of the classical ways, but of course now we do with mass spec based approaches. And then you can identify the protein, quantify the protein, and you can again come back and bring more of the chemistry knowledge to do much of characterization of the proteome. So there are different techniques involved in studying the proteome, from the classical gel-based proteins to the more mass spectrometry approaches now. All gives you the new leads. Many times you are not even sure what hypothesis should be formed if you are studying a diabetes or the cancer or any kind of big problem. What is my actual hypothesis? So these omic technologies can give you the first step towards narrowing down the molecules and give you hypothesis. Now this is my set of pathway which looks very perturbed, and I want to put all my further studies based on that. Then you utilize some functional proteomics technologies to understand that system, characterize them, look at the drug binding, look at the protein folding and other ways of characterization. So protein microarrays and different biosensors for label-free technology like bilayer interferometry or surface pulmon resonance, these kind of tools are being utilized. And of course the CD and NMR etc. are being used. So separating the complex proteome, and again now what we are talking is, let's say, I'm just giving a practical sense, we take let's say tissue samples of your different cancer patients. And expectation is that you want to get at least 10,000 proteins, right? In a single shot, with one injection of your protein sample which you have digested to make peptides, can you get 10,000 protein? And if you can, then you are getting a picture of that disease with 10,000 protein, which is very unique, very powerful. So how to separate the complex proteome has been the challenge in the field. And people have used different workflows, and more classically they started with the gel-based approaches to separate the proteins, then come to mass spec for analysis. But now everything can also be done directly to the mass spec as well. But eventually you have to do bioinformatics and machine learning to get some more powerful understanding of the biological system. So separating proteins was always the fascination and the challenge. Given that now we are talking about 20 amino acids and different properties of the protein to be utilized. So in 1950s, scientists like Close and Farrell, the electrophoretic techniques to build on the size and shape of these protein molecules and how to separate those using these type of techniques known as the electrophoresis to begin with, and eventually it came with the two-dimensional electrophoresis. I'm sure everybody in the room would have done one is the space gel, and you all know like how you are separating the complex protein after denaturation and after breaking all the disulfide bonds and making them denatured, which is now moving the electric field. But at other dimension separation can also be utilized, which is looking at the isoelectric point of the protein. Every protein, when it reaches to a specific pi value, which is closer to the pH, that's where the net charge becomes 0, and then that can be utilized to identify the isoelectric point of the protein, which was utilized on the immobilized pH gradient strip or the IPG strip, which was being used for looking at the separation in the second dimension for the IS. So now, based on these two methods of looking at molecular weight separation or isoelectric point separation, these scientists from 1950s, they devised this method. Can we separate them first in the based on the IEF, isoelectric point, and followed by the molecular weight and SGS page. And every spot which is shown here is now going to give us idea for every protein 2 properties. For example, what is the PI value here? If I start from 3 to 10 isoelectric point, and let's say molecular weight markers are here, I can directly get information this could be around 90 kilodalton protein with the PI of 4. So you already started separating the proteins based on their some properties to begin with, but this map does not give you ability to know the name of the protein. So still you have to size the spot from this particular gel, and then you have to do in-gel digestion, digest the protein to peptide form, and then sequence to get the protein's identification. But people have also started utilizing this technique to compare control with disease. For example, if you can take condition A, condition B, and you are separating them first on the basis of the isoelectric focusing IEF, followed by the SGS page for molecular weight, you can now stain the gels with different type of staining methods, and after that you can compare images and look for the actual statistical changes happening, and based on those you can then now excise the spots of interest which is showing overexpression or down regulation of the specific protein, and then you go to mass spec. So this has been worked quite long actually. I would say this technology has really enjoyed the time till almost 2000 or so. Even many labs across India still are using it. It's not like obsolete technique, but now the amount of labor which is being utilized to get the information which is quite minimal is much more. Therefore people have started moving more to a direct mass spec approach, but this remains still a good way for you to understand biochemistry of the proteome and take your first step forward. How good your protein extractions are, how good your ability to separate the at least 2000 protein on the gel to begin with. You may not get 10,000 right away, but at least to get started. But then the idea was how to identify these spots and get the name of the protein from them. So people cut those bands, do the de-staining to remove the stains, then add the reduction step with DTT, alkylation step with hydrostamide, and then add the enzyme like trypsin or chymotrypsin or IC, so that now you can digest those complex polypeptide chains to the small peptides, and those could be now sequenced with the mass spectrometers. So I hope we are now on the same page that you can take variety of samples, you have different abilities, but now you want to identify the proteins and quantify the proteins using mass spectrometry. So that's where I'm going to now show you some techniques which can be utilized for identifying these proteins. So mass spectrometry is one of the approach which is now being utilized heavily in most of the labs across India, you will see, which is being used to study the protein identification and quantification. A typical workflow of the proteome for analyzing the complex proteome is start from any biological sample, like she wrote you the cell line for example, and anybody was the tissue or urine or blood sample. In whatever sample you take, once you have identified the entire complex proteome, you want to digest them. That is going to be the common step for everything. As I said, the first part is the biochemical, which is your skill of how good you are at extracting the protein out. After that, just all the steps are very mechanical and straightforward. Everybody has to follow the same steps. So then you will take the protein, add the enzymes to make efficient digestion of the peptides. These peptides are in the solution form, and in those solution form, you can't get them to move inside the mass spectrometer. So you have to first make it into the ionized gaseous form, and then you are going to separate these complex ions based on the mass and charge ratio, and generate this type of chromatogram or the mass spec spectrum, followed by you can identify the protein from beta basis. So in any mass spec, you have different nut and bolt, different pieces which you are bound to learn. First is looking at ionization source, how to ionize the protein. Second will be the mass analyzer, how to separate these complex peptides based on the mass and charge ratio, how to detect this finally, and then utilize that information to further search the protein. The scientists which got the credit for doing the ionization especially for the soft ionization technique, to look at the protein characterization using mass spec. Fenn and Tanaka, they got the Nobel Prize for the discoveries on electrospray ionization and matrix-assisted laser desorption ionization (MALDI). And that was one of the, mass spec was already in use. But at the same time, many things are happening very, very good. Like genome sequencing project got completed and we got soft ionization techniques like ESI and MALDI. And that immediately triggered the route for the proteomics research. Before that, mass specs were there, but more for organic molecules and small molecule sequencing, but not being used for the protein sequencing. So now what we do here, you take your peptide, which is a solution form, and that solution form peptide comes from this stabilization cone, and high voltage, you are now making them to the, which is now going to create multiple charge states, and in nice form. Looking at least two type of mass analyzers being used. Right? So any type of mass spectrometer which is having at least two mass analyzers can be utilized for sequencing. For example, QTOF. This complex term, but I think now you can start breaking them down. Okay, what is the ionization source? What is the mass analyzer? What is rejection? So in the mass analyzer, this means, so for example, all these ions are coming to the first collision cell, and then they are going to look at these precursor ions, which now we will, like mass spec will select. I cannot select all the million at the same time. It has a limitation. Let's say very fast, few milliseconds, it is able to read maybe 20 very intense precursors. So you are telling the instrument already, pick up the most intense 20 whatever you can see, and exclude them separately. Then take the next 20. Likewise, you can keep going to the next cycles, right? So now in this way, let's say this particular ions are getting now fragmented in the collision cell, and that's going to generate your MSMS spectrum. Likewise, they will keep doing a corrective way and will not repeat to the same abundant times which you have already seen. So at more of your, your fundamental level of the polypeptide chain and the peptide bond level, now you are looking at the collision energy which is breaking this, the peptide bonds, CONH bond, and it is generating the B or the Y ions. Despite all the fancy mass specs, it will all come to this concept of doing the tandem mass spec and looking at your B and Y ion for all research. So now what you're looking at, the ions which are towards the amino terminal side, there you are labeling them with the B1 ion series. From the carboxy terminal side, you are labeling them with the Y ion series. And given that if you most of the time use the enzyme like trypsin, you will always have, from the carboxy terminal which they work on with the lysine arginine, you will always have much more intense Y ion series in your data set, which you will be doing soon. Then you will see like, okay, yourself that, okay, you have very intense Y ion series and only few B ions which you will see. But those will be utilized for search. So again, some practical name of the instrument like might be TOF, for example. There are TOF1 and TOF2, and you are doing the same tandem MSMS, selecting few precursors, fragmenting those, generating those daughter ions. And in this case, in the TOF, you also have the reflector ion, which improves the separation with the increased path length by using this type of mirror. Another mass spec is triple quadrupole mass spectrometer, which is a misnomer. It does not have three pairs of the rods, it has only two pairs of rods, quadrupole set 1 and quadrupole set 3, in between the collision cell. This instrument is being utilized to look at more targeted, specific peptide sequences. For example, you are only interested in a few peptides to separate. You tell the first collision cell, okay, only going to take those ions, and now going to further separate and generate the MSMS spectra. So again, what you're looking at, same principle of mass spectrometry. You are separating these complex ions within mass and charge ratio and separating them different principles. One was TOF principle, time of flight. Now you're using quadrupole principle. I'll show you now a few more in the our actual lab environment. I visit my thing, I can talk to you. So what you see in the back here, this is from our mass spec facility, one of the Orbit fusion mass spectrometer, which is the tribrid mass spectrometer. It means it contains three types of mass analyzers: the quadrupole, Orbit trap, and ion trap. What you see from the front, those are actually very, they're the black boxes, right, eventually. But I'm just showing you right now is the NGS instrument, which is used for the target sequencing I talked to you in the first lecture. I'll quickly go to the, this Orbit trap, which is one of the still very good workhorse for the mass spec. So again, you're utilizing three types of principles: quadrupole, Orbit trap, and ion trap, how to separate these ions. And depending on the mode in which you run them, or different type of collision cell which you can use, for example, high collision density cell or HT or ETD cell, you can look for the PTM changes or different type of global changes. So again, this part, if I just pause you for a second, since we are doing, this part, which is the nano LC, particular part too, so that based on the reverse phase binding, based on the elution with the gradient, now you are going to separate them slowly, eluting out all the peptides in the column, which is now if you move to the instrument, this part is where the electrospray ionization happens with the needle, and then all the ions are going to move inside this particular black box, which is the mass analyzers of the three type of mass analyzers. Another instrument which I am showing you now in the back is the triple quadruple mass spectrometer, which is, I just spoke to you in by principle, you are looking at specific peptide sequences and targeting those to get their quantification. The different principle involved, you are looking at only quadruple based mass spectrometry, right, which is mass filtering. Additionally, we also utilize this instrument, which is the executive auditory trap, which can be utilized to do the proteomics, but we utilize more for the metabolomics application. And for that, we have used this particular high flow rate setup, which can be utilized for the the small molecules and the metabolites more. So LCMS is very powerful to give you from same sample all the protein data or the metabolite data. And but there are different mass specs. So again, like it's not one instrument which we are talking. There are different principles involved, and depending on application, you choose the different type of instrument configurations. Right? And then the rest of the lab is having other variety of the instrument. This instrument is what you see is the protein microarray setup, which can use the high density protein chips to look at the proteome-wide, locus-wide instrumental and the SARS-CoV-2 proteome arrays based work. And then we have a variety of other technologies in the lab, especially liquid handlers, FTIR, etc. A lot of these clinical samples which we do research on them. And then different server for data analysis. So you are welcome to visit us sometime and then do some experiment yourself. But I just thought to give you where things are, but where things are really developing every day, right? So now Thermo Fisher has brought this new technology known as Orbitrap, Ester Orbitrap. This combines told you, we are looking at the most intense precursors, right? So mass spec is scanning them all the ions and looking at the most intense ones and picking those up and trying to fragment those, right? So this was data dependent acquisition. You are looking at only selected ions you are fragmenting and you are trying to generate the MSMS based on that. In DIA, the approach was different. All these M by Z window comes from 300 to 1400 M by Z. Now take all these window and few precursors from each window, from all isolation windows, and then go next duty cycle and again pick up from all these isolation windows. So DIA approaches got changed. You're not looking only the top abundant precursors, but you're looking at all different isolation window of M by Z. And then, in addition to this approach here, which we are running for every sample, here we also make the libraries. So for example, if you are going to work on 100 samples, you first, let's say, make some pool of these samples, and we are going to run every sample, let's say for long gradient, with very small M by Z, let's say 400 or 500. Same sample, now you will run another time 5 to 600 M by Z. Third time for 6 to 700, and likewise multiple times till 1400. So what in a way you are doing, you are making very high resolution a spectral library of each M by Z in much more detailed way, right? It means it's going to give you very macroscopic views of each of those M by Z. And that is going to make a library. And then once you do the more microscopic quick examination of the DIA, but you put that database for search, that library is very high resolution, so it's going to give you much more precise ways of picking up those ions. And therefore, DIA has, based on these different isolation window, it has overcome lot of missing value data information which we acquire in the DDA based approach. So it within DDA, just let's simplify it. For example, if we have 10 samples, 5 control, 5 treatment. You would expect for a given protein, like there is a higher expression of that particular protein in the disease state. 5 are like in the control are very low and the disease very high. But what is actual situation? Out of the 5 control, maybe only 3 times you see that protein coming. 2 times mass spec is not able to detect that protein. And in disease state, only 2 times it came and 3 times it did not come. So now we found this protein is only 3 times in control and 2 times in the disease. Remaining values are all known as missing values, which is the sometimes could be biological, but most of the time. So to overcome this problem, which was a big problem, so in a way, if you are now going to say, doing data analysis, okay, I'm going to drop all my data which is missing value, you are going to drop 60% data like that. 50-60% data will be automatically down. Many times people say, no, I can't like this. Impute some artificial numbers to impute in those cells when there is no data, which is known as missing value imputation, and based on that I'm going to still proceed with my analysis. Again, all of these words are going to come to you, these terms are going to come to you when you will be doing analysis. So you look at, first normalize the data, look at missing values and impute the missing values, but most of them are very challenging to really mimic the actual biology, because in actual sense it may not, and you are the value which you are getting shadowed with the DIA based approaches. And we first time again brought in India this method, which I had to push my own team to really apply the new methodologies. But based on that, we immediately saw the big change. We used to get around 2,500 to 3,000 protein from the brain tumor tissue. We right away jumped to almost 5,000 protein, and with very minimal missing value data. So that was a big change happened, which with these approaches. Now more recently, we got the timsTOF, one of the very advanced technology, and I'm now showing you some actual data we generated in last week. When we look at EDA-based approach of the one of Thermo Fisher, which is what you actually inject for any mass spec experiment before anyways, and very minimal injection of 200 nanogram when you do. So with Orbitrap, we are able to get almost 2,400 protein with the duplicates. With the timsTOF, we could get almost 3,800 protein. But the major difference here is this Orbitrap we ran for 2 hours, 120 minutes, and timsTOF we are running only for 44 minutes in this case. So time has reduced drastically, but still the coverage is very high. But it is still DDA. Now when we jump to DIA, the difference is much more. And now DIA, even the Orbitrap can give us, as I said, like 4,000, 5,000 protein. This case is 3,400 protein. And now with the timsTOF instrument, we are getting almost 7,000 protein, right? And again, the gradients are very, very fast. Amount of protein what we are injecting is very low. So think about how proteomics is really evolving, right? So I really wanted you to not only know the basics, but also try to start thinking the most cutting edge methods, and the best approaches to tackle the proteome. So if you are the first one in your lab to start with it, don't go with the rudimentary 20 years old approach. Start with something which is really the buzzword, cutting edge in the field right now. Start with DIA, data independent acquisition approach. First prepare samples, digest them, don't go with the germs, directly move to the LCMS approach of DIA. And of course, depending on the instrument availability, either to the Orbitrap or Sunstop or any instrument, but like if you have access to more instruments, choose the instrument which can serve your objective much better way, right? On number 12, we launched this technology in the inaugural event of proteomics. And during a live workshop of almost 800 participants we had challenge of how to give the hands-on to people, right? So we thought to show some live sample run. Live means actually injected in front of you on a video, but the data acquisition was happening on the screen. And we set up with this timsTOF a gradient of 5.6 minutes. Imagine we are talking about proteome coverage in 5.6 minutes. It used to take us days time to now a few hours time, and now it took about 5.6 minutes. In a live audience, it was within 40 minutes, 3 control and 3 treatment of one of the cervical cancer cell lines. Actual cell line were run. And now this is the actual data, which is having some replicate issues, but still almost 2,000 proteins is seen across. And then participants were given the actual data coming out of this instrument to analyze. So think about how the technology is becoming very, very rapid, right? And lot of improvement has already happened, which can, so again 2000 number is not so great for the entire proteome coverage, but think about 5 minutes if you're running something and you're getting 2000 plus protein, I think it's quite remarkable. Again, I had not thought of even a few months back. So again, the proteomics is really rapidly evolving, so look at the newer methods, new principles, new approaches, new workflow, which is going to make you much more prepared. Especially the new softwares are coming, which what we look at in the mass spec, we are always looking at only the peptide sequences. We put together and then try to go database research, and we say, okay, 99% possibility this is a protein. But now the new technology are coming, which are doing the proteome sequencing. They are very much challenging, but there are at least 4 or 5 of them. There's a good prototype coming up with the quantum side, Nautilus Glyphate technology and Oxford Nanopore technology, which are now multiple technologies are coming up with very, very low cost devices, which makes a lot of optimism. In the next 5 years we'll see again paradigm shift of the new instruments and the race in the field, how to sequence the things but more accurately and much cheaper. The way what you see in the genomics is going to happen in proteomics. So I think that gives you like a lot of the stage and excitement. A lot is happening with your proteomics. We have done a lot of work in different disease areas, which are cancer, but I thought not to make a research talk because many of you are coming for GIAN course in the course to start your foundation. But since we are talking about now the tools, I'm just going to give you some glimpse of the new tool which we are developing as well, which you can utilize in your own research. For example, if I talk to you about cell surface protein, and there's a field known as the cross-linking mass spec. How to take the cell surface protein and look at the conformational changes happening. Maybe those conformations are making the cancer cell different than normal cell. It's not just the protein expression level, but the way the functional proteins conform the changes, that might be more relevant. So that's what Dr. Arun Bitta's team has been doing in the leukemia models. He's one of the physician, clinical scientist at UCSF. And when I was doing my sabbatical there, we were looking at this kind of cross-linking data, mass spec data. And the problem was like, what huge amount of data which you generate after different type of cross-linkers, there was no software available to analyze this much more automated and fast manner. So very tedious laborious process, working in the Excel sheet, what postdocs were doing. Then along with our IT Bombay team, with Arun Bitta's team, we developed this Alpha Cross Excel software, which is a Python-based program, which can utilize now the AlphaFold for structural information. We take the sequences from our actual data, FASTA data sequences, and then also utilize the PyEvol and the Prodigy for looking at different residual distance calculation and showing where these crossings actually being formed in the protein and show them in the PyEvol visualization tool. And based on that, we took different data set, different models. In fact, Thermo Fisher team from California is also working very closely with us on this project, where now we have tested many data sets. This software is under review right now, but will be made publicly available soon, but already featured in the ASMS conference. So again, it gives very good prediction with the AlphaFold, with the HLPDB structure, and it's one of the way of looking at the more cell surface and the crossing mass spec data. While doing a lot of work on the mass spec based proteomics of the brain tumors, We were generating a lot of data, but we always had challenge of getting the right controls. And therefore, we thought, okay, should we look at systematically different type of normal brain region? So with different proteins are expressed very differently and at different levels, which is expected, but there was no map being made before that. We also developed the brain phosphoproteome map of the different regions, sub-regions. And all the data was very well appreciated by the journals. And JPR published in the cover page, and the editor invited us to make a database for this to make it publicly available. And we thought to make it publicly available. But then the COVID thing happened, and that gave us an opportunity to have hundreds of interns working with us online. And many conferences when I go and talk about it, at least somebody has contributed, which I have now almost 56 brain diseases, which one could get a lot of information for the brain disease atlas and look at the transcriptome map or the proteome map, even the drug finders, or looking at the marker curator. Again, Professor Graham has been a good collaborator to look at machine learning approaches which can give us some better ranking to identify the markers for different diseases. So again, these are all good tools for you to, if you work in the neuro research, neuro area, we should start exploring some of these tools. More recently, which is work being currently pursued, we have developed the drug AI, which is again, so much work goes on as you were talking about, doing so much experimental work on the drug. So much work goes on as you were talking about, doing so much experimental work on the drug, and then you do not know the drug prediction that is going to really work or not, this protein. So we utilize different type of available resources first of all, and what all information of the protein is structural can be utilized to make the better prediction for a drug is going to bind, or the protein is druggable or not. So we also looked at all the FDA data set, which drugs have been approved, which drugs are under current trials, and which are going to be for different testing being done. And based on then all this data set which we acquired, and again huge number of undergrads from IT Bombay without PGs were involved, then we utilized different type of machine learning models, from the random forest to the XGBoost, and build those models to say, okay, now we are training the model. FDA data set was used, approved data set was used to train the model because they are approved drugs. Right? These proteins are already approved. It means they have these machine learning algorithms, and if they are able to do well, then we can utilize them for predicting the unknowns. So this will be developed in March this year. But then, since then we were under observation to see okay, from March till November, which drugs, which proteins were approved by the, so that time they were under clinical trial, and at least 9-10 of them which got approved, we already had our own data prediction from that, which matched almost 95%. That was showing the power of these predictability tools. So again, this is going to be made publicly available soon, but a lot of prediction data which I'm showing you here is confirming that we can now predict the druggability index of these proteins ahead of time, and you can save a lot of your time and your valuable energy and cost for doing these experimental models. So this will be available very soon. So again, currently under review. So okay, now you can see in which way experimental data complemented with various type of these tools are going to improve our pace and ability to understand biology better, right? Additionally, we were also part of this human proteome project to look at the draft human proteome map. The first proteome map was published in 2014-2015, but there was only 70%. In the COVID time, Mark Baker from UPO led this project, which we were part of from IT Bombay, now gave us almost 91% draft human proteome map. Again, a good database, experimentally data available, which you can utilize for searching against the proteins. Again, these are all ongoing efforts which improve the community research in a way to improve our precision and ability to do the proteomics research. More recently, just 2 days back, I came back from China, where I'm part of this big Pi Hub project, which is a multi-billion-dollar project funded. Almost 30 scientists from out of China have been invited for this meeting. This work is going to be out shortly in Nature, where now first time there's going to be the human navigator for different organs and different type of proteome to look at. And idea is to utilize that towards more broad scheme of wellness, not only just for the disease scheme. Watch not only our own work, but many other like MS Fragment and all, and use them for your own research, because the things are the way AI is developing now, things are much faster, right? It has to be accurate, but it is very fast. You have to keep up the pace, right? So therefore you should be very open learner to learn new tools and utilize them, and if it works for you, embrace them for your research. With this, let me thank all my team members for who are contributing in different projects in the research lab. Many collaborators from IT Bombay and other parts of India. Many funding agencies which are supported. I can't name them individually, but they're very generous from India and international to really help us in a variety of ways which we have been doing the research. Again, some of you might be, after this workshop, might feel more prepared to go to the next round, and you might want to learn some more proteomics. So we are planning to launch this internship program. Alright, so this will be Proteomics Advanced Winter School for a month, which will be December 10th to January. And since I get a huge number of requests from students to do internship and the projects, which typically I can't take that many people, we are always very poor number of people in the small space that we have already. So I conduct these programs more in the hybrid mode, which is very convenient for anybody to learn from anywhere. And we do a little bit lab visits as well as a part of it, which is not mandatory but can be optional. So the way we do it, every day some lectures will be released. Every week we do more online interaction to clarify your doubt. Every week is a quizzes, and during the entire 1 month, 2 times we will have visits, especially people around Mumbai, Pune and those regions, they usually come. So some outside, even sometimes they want to come IT Bombay, they can come. So this could be a good opportunity, and again many people want to do 4 months, 6 months long project with us. And again, I can't just take blanket huge numbers. So those who do well in these things and they are well trained and they are motivated, we can again shortlist them depending on their requirements for the long-term projects as well. So this could be, at least for some of you, maybe opportunity. But for many of you who may not be interested in internship, should definitely come to IT Bombay in February for a big proteomics gala event, which will happen. Almost 14 workshops will happen simultaneously with the big international Advances in Proteomics Technology Conference, February 14 to 17. So block to date, even this year we did, which was a big success. We got all the big scientists of proteomics. Dr. Roop, Minister from Allahabad University, they also came. Almost we had 2,000 people who were part of that event. So we'll be happy to see many of you who are building your curiosity and knowledge, and maybe opportunity to directly interact with the best global scientists and explore more opportunities. That's all. Thank you very much. I hope I gave you some knowledge and information. And I hope I have not taken them too much time, although I did.
So I think if you have
we can talk about the lecture, I'm happy to have more interaction. But if any 1 or 2 questions you want to...
1 or 2 questions if you... Maybe even about the internship, how to approach it. I was also I joined through an internship training program. That's how we...
Well, very proud to see you, now you are...
so in 2019, I was attending a training program at IIT Bombay.
It's always like that. In this area, I always feel myself a student, right? As I said, when sabbatical, when I went out there, my approach was to pick up the new technology, Again, in India, one thing, we always feel we are situated now and we know everything, right? So, but it's so rapidly growing field that you can't be, expert of everything. New softwares, new tools, new workflows, new instruments. So to really keep up that pace, you have to be a modest student, a good learner all the time, right? So I hope at least, it gives you a primer, a motivation to read more, to understand this field more. And of course, a lot of my videos are available online to get more in-depth lectures about it. And I will be available as well with Dr. Roop's things after this workshop to talk to you more. Okay? All right, enjoy your lunch. Thank you.
Okay, so participants, we have the lunch. You can have it in this area, right? Don't bring food into this room. That's one request. You can have water. Don't get food items in the seminar room. And if you have any issues, just contact our volunteer.
पूछते हैं इसको ऐसे ही रहने लाइव देखते हैं अरे वो साइड डिजिटल इंडिया वाला चल नहीं रहा है उसकी से सारी दिक्कत हो रही है कौन सा ऑप्शन है
डाटा का अगर वाईफाई एयरप्लेन मोड पे डाल देते हैं तो उसके बाद भी नेट चलता है
ऐसा कुछ नहीं होता
है रेडियो पे अरे ऑप्शन
लेकिन मैं क्या करूं लाइफ में पहली बार हम सुन रहे हैं
नहीं होता कर चुके हैं मेरे वाले में नहीं होता
मेरे वाले में चेक कर लो शायद से हो जाए